QuickTest ELISA kit. Preparation before the assay. This video will show two-step ELISA assay procedure for rapid detection of human IL-ba. Equilibrate the ELISA kit for 20分钟 at room temperature. Check whether all components are available according to the list in the manual and then open the kit. Core kit components: lyophilized standard, sample dilution buffer, cap, DAC working solution, HRP streptavidin orange, concentrated wash buffer, 25x, TMB substrate, stop solution, ELISA microplate, dismountable, plate sealer, manual. Preparation of wash buffer. Heat the wash buffer containing crystals at 40 摄氏度, over 50 摄氏度not allowed, via water bath till complete dissolution. Preparation of standard. Centrifuge the lyophilized standard tube at 1000 克 for 1分钟. Add 500 ml sample dilution buffer into lyophilized standard tube and tighten the cap. Gently shake several times and immediately mix by a vortex mixer. Then, centrifuge and collect the liquid to the bottom of the tube. Store the rest lyophilized standard in a sealed bag at 20 摄氏度 to avoid dampness. Take 7 clean EP tubes and add 150 ml sample dilution buffer into each tube. Then, take 150 ml standard solution and dilute, at the ratio of 1:2 respectively to get the gradient concentration, 1/2, 4/1, 1:8, 16/1, 32/1, 64/1 of standard. Sample loading and incubation. Take the required strips and put them on the microplate. Put the rest strips together with the desiccant into the sealed aluminum foil bag and store at 20 摄氏度 to avoid dampness. standard and sample loading. Add 50 ml antibody working solution into the relevant well. Notes: add the solution to the bottom of the well without touching side wall and foaming. Then add 50 ml standard or sample immediately. Notes: tips should be replaced during adding another sample. Gently shake the microplate for 50 seconds and then cover the well by a plate sealer. Notes: for sealing, sealing should be tight to decrease the edge effect and CV differences caused by evaporation. Incubation: incubate for 1小时 at 37摄氏度。Washing: remove the plate sealer and discard the liquid in the well. Then, suck 300 到350 wash buffer from the clean loading. Slot with the multi-channel pipette and add into the plate well to wash. Discard the liquid in the well into the waste container. Immersion is not required. Repeatedly add 300到350 wash buffer for a second washing. Remove the residual wash buffer by gently tapping the plate 2 到3 times with your hands holding both sides of the plate. To avoid overdrying, add the reagent required for the next step immediately. HRP streptavidin and incubation. HRP streptavidin is orange and ready to use for directly adding. Put suitable amount of HRP streptavidin into clean loading slot, then rapidly add 100 ml working solution into each well with multi-channel pipette. Single-channel pipette is slow and will cause high CV or negative. It's suggested to perform the assay after well preparation. Cover a new plate sealer after adding HRP streptavidin. Incubate the sealed plate for 30分钟 at 37摄氏度. Take TMB substrate, stored at 4 摄氏度 out of the refrigerator and equilibrate at room temperature. Washing: remove the plate sealer and discard the liquid in the well. Then, suck 300 到350 wash buffer from the clean loading. Slot with the multi-channel pipette, add into the plate well to wash. Discard the liquid in the well into the waste container. Immersion is not required. Repeat the washing process 5 times. Remove the residual wash buffer by gently tapping the plate with your hands holding both sides of the plate. TMB substrate and staining. Take suitable amount of equilibrated TMB staining solution according to assay requirements. The loading slot of TMB substrate should be clean and disposable without touching HRP enzyme. Before use, wash with deionized water to prevent the pollution of TM. Add 90 ml TMB substrate solution into each well. Usually, the standard curve well will show the blue gradient. Blue also appears in the sample well with higher concentration, which is not caused by the ELISA kit. The staining of standard curve should meet the criteria first. The valid data of standard curve can accurately calculate sample concentration. Notes: don't pour unused TMB into the bottle. Statically incubate the sealed plate for 10到20分钟 at 37摄氏度 in the dark. The staining duration is about 15分钟. During the staining, observe the color depth as well. Properly control the staining intensity and duration to stop the reaction. Please refer to accurate TMB visualization control specified in the manual. After stopping the staining, take out the plate. The staining duration can be extended or shortened accordingly. When obvious blue gradient appears in the standard well, the reaction can be terminated. Termination of staining and reading. At the end of staining, add 50 ml stop solution into each well. Then, the color turns yellow from blue. Notes: the order for adding stop solution and TMB substrate is the same. Reading of results after adding the stop solution. Immediately read the OD absorbance at OD 450 nm with a microplate reader. If your microplate reader has the corrected wavelength, set 570 纳米 or 630纳米, the corrected value 等于 OD 450, OD 570, OD 630. The correction can remove the OD value of non-staining substrate to obtain more accurate detection result. For the microplate reader without 570纳米 or 630纳米, use OD 450.